And would like to invite Valtencir, Zucolotto,
to give a speech, Nanomedicine, new aspects about nanotoxicity. Valtencir is of the Physics Institute of São Carlos. Thank you very much to the coordinator of the roundtable. Before beginning, I'd like to thank this opportunity that was given to us to come here to speak to such a distinct audience, and thank Mr. Professor William Weissman for this kind invitation. We have exchanged lately experiences and ideas. So I would like to talk today about nanomedicine. I think that, slides. We were talking about this blink problem. I think we'll have to use the pen drive because we will. Have been already talked about in this brilliant, previous brilliant presentation. We're going to talk about our experience in the production of systems of diagnosis and therapy using nanomaterials, and also see some aspects about nanotoxicity. As an outline, we're going to talk about the union of these two areas, the frontier between nanotechnology and medicine, and talk about nanosystems for diagnosis, nanomaterials for tissue engineering, and nanoparticles for cancer therapy, and nanotoxicology. My first phrase would be, from the medicine point of view, nanotechnology is a very good thing. If there weren't nanotoxicity in some aspects, we would not have almost no application of nanotechnology in medicine. Very quickly here, the majority of all of you here have a familiarity with the theme of nanotechnology. Everything began, for those who do not know, this became with Richard Feynman in 1959. He was the first one to perceive that the scientists are losing a lot of space when they are processing matter. So they had his famous phrase, "There's plenty of room at the bottom." So in this his speech in Caltech in '59, he said, how can we write the 24 volumes of the Britannica Encyclopedia in a pinhead? In a physics and chemical point of view, nothing could make us write this. But at that time, now what we know is that we don't have the pen to write all this information. So what is important is to process these materials at this level. So this is where the boom and the revolution came in nanotechnology. In the point of view of the material engineer, nanotechnology came to produce nanomaterials with new properties without having to synthesize another new material which costs a lot. So processing a material using a nanoscale, you have much better properties of the new material. For example, gold in the macro scale, it can become liquid in 1,000 degrees Celsius to reach to 5 to 3 diameters of nanometers. It will become liquid in 500 degrees centigrades. So the surface of this particle will be linked together. And this is the specific superficial area. It will reflect the light. The gold in nanoscale is red. It changed completely. It is how it interacts with light. So this is nanotechnology. This is where the infinity of the fantastic, enormous applications in which we can use. In one of these fields, maybe the first one, and actually the most benefited in the nanotechnology, is in the medicine area. So what I present in this slide is a scenario of medicine, where's the interface of nano with medicine. So we have three big fronts, basic fronts. We have nanomaterials in medicine. The first one in the diagnosis part we just saw right now in the speech, the diagnosis of cancer. And so the biosensors that are the using molecular imaging and tomography, CTs, where the contrast in the image is amplified by the presence of the nanomaterial around the tumor of that tissue that could not be seen in another way, and using genosensors that only with this we can see and detect small anomalies of DNA with a low cost. We can use the PCR for this, but with what cost? It costs a lot. So this is the genosensors has this advantage. Another front that we have is the regenerative part. Kawi has commented about things that Nanox has been doing in the part of the processes modifications that receive a nanometric treatment, and the way this processes talks with the tissue to be able to acquire this capping, and also the cell scaffolds with the therapy with the stem cells. That is something fantastic. But if you don't have a matrix to transport and transplant them to the human body, nanotechnology has worked a lot. And the part of therapy, we have some fronts that are very important: the drug delivery, the photodynamic therapy, hypothermia, and what we call the intrinsic toxicity with some nanomaterials. Some of these therapies occurred before nanotechnology, like photodynamic therapy. in this interface where we're dealing with human lives and utilizing new materials in live organisms, we have to take care with the nanotoxicity, and a lot which is will be worked with the nano agro-business. So in a country like as ours that has agriculture and that has a positive balance in our economical budget is very important. This is an overview with the principal materials that we have in our laboratory in São Carlos. I coordinate this laboratory. Here we have some samples of materials which we have produced very easily and quickly, and they produce these nanoparticles, metallic nanoparticle of silver, platinum, gold, and these are the images and the photos of these nanoparticles, carbon nanotubes, and ketosane nanofibers, polymeric nanoparticles that we use for drug delivery, and nanoparticles that are linked to antibodies and proteins, and they are very important in the industry. So this is just an overview of what we have been producing in our laboratory in a simple and relatively low-cost way. For those who have never seen the synthesis of nanomaterials, one of the roots, the bottom-up process in the chemical synthesis is extremely simple. We have this nanoparticles of gold. We begin with a salt of gold, which is not expensive, and it is in a solution. And when you put the reduction agent, you become with the ions of gold, and the particle will grow, precipitate, and fall in the beaker. Before, we put a template so that it will not make the particles grow, and they will be stabilized, and then you can produce nanoparticles like this one with an excellent quality of size that influences everything in the nanoscale. So it is simple and relatively cheap. Let's talk about nanomaterials for diagnosis. Today, all the nanomaterials in lab scale have been used for physiological, biological something. Carbon nanotubes, dendrimers. The principal analytes are glucose or urea, uric acid, LDL, neurotransmitters, adrenaline, serotonin. So I'm going to get some of the examples in our laboratory and talk about some of these applications. In this first case, we're going to show a nanostructured sensor where we organize these particulars of gold in the dendrimers. Here we have a nanometric film, and where we mobilize an enzyme, oxidase glucose, and we will put it in one of the those tapes in the like the glucose sensors that are sold in drugstores, and we obtain the detection of glucose in a very low level. The glucose that you want to detect reacts with an enzyme, and in this process, two electrons are produced, and these electrons have to go to an electrode. But in this case, they have a metallic nanoparticle, and they go through them, and we can detect efficiently the response to this. As I want a conducting medium to see how many glucose molecules I then we're going to use some nanowires, metallic nanowires, with cobalt, nickel, and molybdenum. Here we're going to see the edges of the nanowires, and over them we'll put an enzyme, and above it I will put also an acid. The glucose doesn't have a negative charge, so it can go through this layer. It reacts with the enzyme, which will be detected by the electrode, and I'll see the current and the concentration of glucose. Where we have this glucose, we have the ascorbic acid. And then I have to see that I'm detecting the glucose exactly. So this acid has a negative charge. So where I can only see this with the nanostructure of my electrode. Another one are the nanotubes of carbon nanotubes, and we make it in alternate layers with a molecule that are the phytanocyanides. It has a big affinity with dopamine, for example, and using this same electrochemical configuration, they are very blue, and we have these proteins in the tube, so we can see the variation of the current with the concentration of dopamine. We can detect the quantity of dopamine that is in that moiety. Third part, I'm going to talk about some sensors that we're using for diagnosis of illnesses. One of the first systems that we used was leishmaniasis, which is a big problem in the countries, developing countries like Brazil. There are many people who live in risk areas and they have a serious problem with diagnosis. Many times you have to wait for the clinical manifestation of this illness, and you have to make a culture, cellular culture, to see if there is the protozoary there. Sometimes it's not feasible. But if a person has two illnesses at the same time, like Chagas and leishmaniasis, it will be difficult to detect because the epitopes of the antibodies are very similar. So we developed with this, with Ribeirão Preto, the researchers from them, a system that is efficient and cheap. In Ribeirão Preto, they isolated the proteins of SAPE, and these antigens of these proteins were used with the liposomes that were called proteoliposomes. Here we have the cartoon and the real photo. These proteoliposomes that have the antigen, they were immobilized in the surface of an interdated electrode, and we tried to detect the presence of antibodies against leishmaniasis. If the person is contaminated, they have antibodies. You can identify it with the antibodies. So quickly, the physics behind this. How can we detect this bond with the electrode? Here we use the electrode with alternated measures. If this electrode has two capacitors, if my capacitor, after I mobilize the antigen, I have a certain sign of capacitation. If I have a solution that has the plasma with the antibody, and this this plasma links itself with the electrode, then I can assess and see how much and if I have leishmaniosis. For an electrode without nanometric coverage, if I work with different antibodies, I will not see this capacitation for any frequency. But if I have an electrode with an antigen and I merge it in different antibodies, I can process it, and I will have cartoons like this one. This is my output. All these samples that were I will have the clusters, different clusters, and see the contamination, because then I can relate this signal with the quantity of antibodies that I have. If you can detect leishmaniosis today, but if it's a case of an epidemic, you have to make a screening of 5,000 people. No one's going to pay. Public health will not pay this. So we have made the patent of this system because it is cheap and discardable to see who has been infected or not, and afterwards continue their health system. Here we can also see that there is an immobilization of nuclear receptors for thyroid hormone detection. You can have excess or the contrary of hormones in the organism. So they were used an electrode to detect 3 hormones, for example. Here we have an analogous, synthetic analogous, and after processing the data of the capacity, we'll see the result. We'll see concentrates that are 2 to 3 nanomolar to 500 nanomolar. And the second part of the presentation, I'm going to see how we use nanotechnology for regenerative medicine. Here we have nanofibers that are of polylysine that are they're covered with other nanos, and we use this as a template and to see with an electric pulse to see if it grows more easily. So we grew in this cells of cardiomyocytes, and we observed that after some time there was a lot more of cells reproduced themselves with the electrical stimulus with the nanostructure. Now we'll go to another part that says, how does the nanomaterials talk to biomolecules, membranes, and living cells, organisms? We have to have a concern with this because we will see therapies with cancer and toxicity. Our immune system is prepared to interact and deactivate things which it knows. 20 years ago, a person didn't have a risk of breathing through a carbon tube, but today, maybe in the carbon tube, this can happen. So we have to learn how to work with this. So there are new things for our immune system, and we still don't know the response of our immune system for some nanomaterials. So we started investigating 3-4 years ago in our lab how this interaction of nanoparticles with biological systems happens, and how they relate. So we had some interesting results. We had nanoparticles of gold with methionine, amino acid. After obtaining this nano spaghettis, nanotubes of where we see the nanoparticles in this tube of methionine. But what is important is the results, that we don't have to be specialists to we have to interpret this. All amino acids have we have the amino group and the carboxylic group. So here we saw the result of the spectroscopy, how one amino acid interacts with another. When you don't have a nanoparticle, or when you only have amino acids, this experimental curve to see how one interacts with other. One interacts the positive with the negative part of the other one. So this occurs when we use the same analysis in the nanoparticle. This band disappears. So what happens? This band disappears. So the one amino acid doesn't talk anymore with the other between its negative and positive parts. So the band increases, which means that they interact through their amino groups, through hydrogen bonds. So what we see here is that when we have a nanoparticle, depending on the conditions, I can change a part of how a biological molecule interacts with another one. And this is what gives me an incentive to start learning about nanotoxicity and also nanomaterials for cancer therapy. This is the positive side of nanotoxicity. We could not think of nanotherapy for cancer if we hadn't seen the nanotoxicity. What we have worked of the gold toxicity that in the liver carcinoma cells with a positive charge, we use the doxorubicin, that is an anti-tumoral that is used in chemotherapy, and we saw that in concentrations of 50 micromolar, which is very high, you can have cell death, and more with the positive charge. We can separate with how many cells died by necrosis and how much died by apoptotic cells, which is a programmed death. The time of incubation is of 24 hours. And talking about an assay of this comet assay, that we call it a comet assay, we see that cell death was also caused by injury of the genetic material. So if I have this tail, that's why we call it a comet, this shows that we have had injury in the genetic material of the cell. So the nanoparticles cause that. And in this therapy, there's another aspect in which we use nanomaterials for drug delivery, and which comes they come from the Trojan horse's concept. You have to take to a part of the body a chemotherapeutical agent. I have some colleagues that can explain this much more. But quickly here we have some nanoparticles like liposomes, nanospheres that can be polymers, dendrimers. Each one of them can keep inside them agents, chemical agents that will fight against this disease. And there are also markers that can specify where they are in the body. the markers can be an antibody that can identify a specific type of tumoral cell. And here we have particles of chitosan with metacrylic acid. They have triac, one of those hormones inside them. In these nanoparticles, we're trying to reproduce for we can mobilize them in very thin membranes to be used as skin patches for the hormone reposition. And here we have a curve of the liberation of this these nanoparticles that has the pH that is equal to the skin's one. So this is an example of therapy. We have patented also with Professor Kawe the production of nanoparticles of silver that are used also with chitosan, and it is less aggressive to the environment, and it has a bactericide activity. 20 micrograms per ml can inhibit the growth of Escherichia coli. So these particles are easy and simple to produce, and I can process them in incorporating them in the fibers of sheets or of patients' clothes, of also the paint that goes on the walls of a surgery room, and to help against hospitaler infection. Another example is where nanotechnology can be very cheap for a very serious problem. When nanoparticles are toxic for tumoral, we call it therapy, but when they are toxic for healthy cells, we call it nanotoxicity. So here we see an example of gold that we're talking about the blood cells with high concentration. So when we see the nanoparticles with negative charge and are not toxic for skin cells, for fibroblasts, at concentrations until 50 molar. Everything depends on the concentration, the size of the concentration. We have also seen a study of mimetizing cell membranes because it has to interact with the membrane. We mimetize an artificial membrane, and we see how the nanomaterial penetrates. And in some studies, we also use mice that have presented after a bit an alteration in the size of the bladder, for example. And what we're doing now is the production of these nanoparticles that are very efficient, and they're very beautiful to see because these are nanoparticles of a protein, and inside they have nanoparticles of gold. This can be used as a drug delivery system. We produce this kind of coating in these polymeric films which contain particles of chitosan with nanoparticles of silver that has a big there are parts of this membrane where the Staphylococcus aureus does not grow, and we could press them in films as films that can be used. We're using works with hyperthermus. We're using magnetite dioxide of iron dioxide, and if they are directed, it increases the temperature and kills the cell. We have begun a detection of genosensors to detect using nanomaterials. We have one that is used for hypertension, and we have a system that has an anomaly, and we have these nanoparticles of gold that are together with the sequence of DNA to detect these anomalies. Who hasn't seen this? We use a platform, an electrode. You mobilize a small sequence of DNA, you complement it, and then you put it in contact with a real sample. If you have this sequence that where I have to detect, and after cleaning it well, washing it well, I will put it in contact with my reporting sequence that is a complementary part that is linked to a nanoparticle. And when this happens, I have a signal. If the nanoparticle is a conductor, I'll see the variation in the electric current or variation in color. So it's a very simple way of detecting this genetical anomaly. To finish the actors of the nanomedicine, we have to see all of them. In the more scientific part, we have doctors, engineers, physics, chemists, biologists. Industry has to accompany this because we in our lab, we cannot produce all these materials, and also the government funding and overall, and also in the regulatory part, some patents that we have produced in 2010. We have produced a lot of patents with the support of the Innovation USP, and this is summary of a study in which I participated in CGE. Professor Tedesco was also there, and with the impacts of the nanotechnology in the health and care industries, which are significant, from the drugs and medications, prostheses, and collective health and hygienization. And I would like to thank many people who have collaborated with us in USP and out of USP. I have helped Don Ivone with one of the networks of the Nanobiomed, and it is one of the CAPI's networks. This is our group of study. They are the ones who are always working. This is our site, if who wants to visit our publications. And our students also have the Twitter and always telling us about everything. Thank you all, and sorry for the delay.